Hey guys, welcome back for our last set of notes for chapter 3. I hope you forgive me last time. I got jumped the gun just a little bit there. It said we were done with proteins and ended the video, and then when I opened it back up to talk about nucleic acids, I saw there were 2 slides left in proteins, so I apologize for that, but we will go ahead and wrap this chapter up here. Why is protein structure so important? Hopefully that question's incredibly easy for you to answer because, like I just mentioned, protein shape determines its function. And I said that's very, very important. Please remember that. So proteins function only as long as they maintain their correct three-dimensional shape. Changing a protein's shape may have drastic health consequences, or it might just change your hair from curly to straight and then back again. You never know. So denaturation. So this is a little bit more severe than like what a straightener or curling irons might do. We looked at that picture of keratin earlier in the chapter, and that girl in that picture had super, super, super curly hair. If she wanted to straighten it, she could use a straightener, and she could use a lot of heat. And in this hot straightening, I don't know what you call them, perhaps an iron or something like that. Maybe you just call them a straightener. In these hot, flat pads, that heat would break the disulfide bridge or the sulfur to sulfur bonds in some of the tertiary structures in the keratin, and those proteins would actually start to fall apart just a little bit. Now, keratin's pretty tough stuff, so it's not like the hair is going to literally physically fall apart, but in the chemical sense, it kind of does. And when that keratin protein, when those bonds are broken, that keratin protein starts to fall apart, that hair loses its natural curly characteristic shape, which was the result of the protein, and will now be straight because the heat from the straightener has changed that keratin. Now, that change is temporary because as the hair cools down throughout the day or 2 days or whatever, the hair will start to reform those chemical bonds, and it will start to reform its natural curly shape. So heat is a big part of a protein's shape, and it can really mess with a protein. Now, thankfully, straighteners and curling irons don't mess with our health too much, so that's not a big deal. But if you expose a protein that does a super important job in your body to severe heat, you change its pH, you make it very acidic or very basic. Certain salts and even a detergent, we can start to disrupt some of these bonds, like sulfide bridges and hydrogen bonds, that are really working hard to maintain that protein's shape. And if it loses its shape, you know it loses its function. And once it loses its function, we call this denatured. So if you lose your shape, you lose your function. And depending on the protein, that can be a very bad thing. So here's a cool little thing for prions, a cool little thing for proteins. You may or may not remember this from Gen Bio, but sometimes there is a disease-causing agent which is not a virus, and it's not a bacteria, and it's not a fungus. It is just a protein. And this protein is misfolded, and it can cause disease. And we have mad cow disease, or BSE. That's caused by a prion. We also have Creutzfeldt-Jacobs disease in humans, scrapie in sheep. Here in our area, we see occasionally a disease called blue tongue, or if you want to, make that into a 25-cent word, it's called epizootic hemorrhagic disease, and that I'm pretty sure is caused by a prion as well. And so, just these animals that are either, it's in their water or it's in their feed, and they ingest it, and they get these diseases. And really, all they're ingesting is a protein. So it's kind of amazing. All of these diseases, these 3 here at least, are characterized by a deterioration of the brain. That's where the spongiform word comes from. The brain in the cow starts to look like a sponge with these big pockets that are kind of decaying, and it's sort of horrible. And physical abilities decrease, and they're most certainly going to die. Yeah, that's what happens when you get giant holes in your brain. Okay, so Here's Creutzfeldt-Jakob disease, and you can see these pockets where this prion has caused these huge pockets of protein and disease that's causing this Creutzfeldt-Jacobs disease right here. And it is a sad thing because, to my knowledge right now, there's not a whole lot we can do about it. Once you have the disease, I think that's kind of the long and short of it. That's going to be about the end of it. So, take-home message, why is protein structure important? We know that. Its function it depends on its structure. Conditions that alter the structure can alter the function. No kidding. And if we get it too hot or too acidic, we can break hydrogen bonds and unravel the protein. And then we could potentially be in really bad shape. So, we've done carbohydrates, and we've done lipids, and we've done proteins. So we're 75% of the way home. We just got to finish our last major macromolecule, and that is a nucleic acid. So nucleotides are the building blocks of nucleic acids. Everything's had building blocks so far, right? Our proteins had amino acid building blocks, and now we have nucleotides as the building blocks of our nucleic acids. So the two nucleic acids you need to know: DNA, RNA. Deoxyribonucleic acid, ribonucleic acid. So it's easy to remember because the name's right there for you. Some of these nucleotides do have a role in our metabolism. So our nucleotides are a really pretty small organic molecule. They have a sugar, they have a 5-carbon ring, and they have a nitrogen-containing base. And that's really the part that gets a lot of our attention. They also have one or more phosphate groups, which will help us link one to another. And then ATP is a super important molecule you're going to learn a lot about. But that is actually a nucleotide as well. And this isn't so much for carrying information like our DNA and RNA are. Our ATP is adenosine triphosphate, and that is a nucleotide that has 3 phosphate groups on it. And he, hopefully you already know him, is the energy currency of the cell. He carries the 3 phosphate groups, and there's a lot of energy in that. So here's what the kind of general structure looks like for a nucleotide. Here's the base. He gets most of our attention, so we'll start with him. This is adenine. They can be a 2-ring structure or a single ring structure depending on the nitrogenous base you're looking at, if it's a purine or if it's a pyrimidine. If it's adenine or guanine, those are purines. And if it is cytosine or thymine, those are pyrimidines. And so here we have our base adenine, and that adenine is attached to our ribose sugar. And so right away, you know if this is DNA or RNA. It's of course RNA, ribonucleic acid. I mean, you could always read down here as well, but that's not nearly as much fun. And then from that sugar, we also have our phosphate groups attached. And so that is a nucleotide of RNA, and it is ATP, the one that they're giving us here. So here's our chain of nucleotides. I mentioned before that phosphate group was kind of how we linked them together. Here you can see our phosphate as that circle right there, linking one nucleotide to another. So a chain of nucleotides is what we call a nucleic acid. That's our DNA and our RNA stuff. The sugar of one nucleotide is covalently bonded to the phosphate group of the next nucleotide. And so what we get is a sugar-phosphate backbone connecting these together. So once we get polymers of nucleotides together, in which the sugar of one nucleotide is attached to the phosphate group of the next, we can start to build these really long molecules called RNA and DNA. So our first one we're going to start with is RNA. It's ribonucleic acid. He contains 4 kinds of nucleotide monomers, including ATP, and it is super important for protein synthesis. So the 4 that RNA contain are cytosine, guanine, adenine, and uracil, because we go A to T in DNA and A to U in RNA. So that is one important difference to note between DNA and RNA, which I'm certain we'll get to later in the year as well, but just so you can kind of be looking ahead. DNA is double-stranded. RNA was single-stranded. DNA is double-stranded. It's 2 chains of nucleotides. They're twisted together to make this double helix, which you're all very familiar with, and they're held together by hydrogen bonds. So there's that famous picture of Watson and Crick looking up at their model, and it's like this twisted kind of ladder sort of thing. That's what they mean by a double helix. DNA is incredibly important because it contains all of our inherited information necessary to build us, and it is coded in the order of nucleotide bases. This is why nucleic acids are so important. So here's what DNA looks like. This is kind of what I told you, that twisted ladder sort of thing. The cell uses the order of the nucleotide bases to guide the production of RNA, and the RNA produces the proteins. And so the DNA is incredibly important to us. So, what are nucleotides and nucleic acids? Nucleotides are monomers for nucleic acids, or building blocks of nucleic acids, DNA and RNA, and a few have additional roles like energy storage. DNA's nucleotide sequence is what encodes our heritable information, a lot of the information that makes you you, and the information that will of course be passed on to your children. It's all DNA. And then RNA plays important roles because the cell uses the instructions that's written in its DNA, then is transferred to its RNA, and then we build proteins out of it. So that is very important as well.